All right, I think we have a great number on board. So again, welcome everyone. I'm Dr. Jhanelle Gray. I'm the chair of the Department of Thoracic Oncology at Moffitt Cancer Center. We are so very thankful for you joining us this evening for this information-packed webinar. I'm confident that you will find value in my colleagues and I will inform and share with you today. Before we begin, I just kind of have to take care of some housekeeping items. So if we can move to that slide, please. This content is not intended to be medical advice, and the viewers should consult their physician should they have any questions. The viewers should not rely on this information contained in this presentation for immediate or urgent medical needs. Additionally, if you think you have a medical emergency, please call your doctor, go to the nearest emergency room, or call 911. Never disregard professional medical advice or delay seeking care because the information contained in this presentation. And to circle back on again, we're so pleased to have you here with us tonight. For those of you who don't know, I wanted to kind of kick things off, just to give you an overview that Moffitt Cancer Center is the third largest cancer center in the United States as far as patient volumes, and we are frequently on the list of top cancer hospitals in the country by US News and World Report. We are really a particular strength at Moffitt. That is the Department of Thoracic Oncology. We specialize in different thoracic malignancies from screening to prevention, as well as diagnosis and treatment, including lung cancer, mesothelioma, thymic malignancies, and Pancoast tumors, as well as other malignancies. We have a multidisciplinary team of thoracic medical and surgical oncologists who all work very closely and collaboratively together with our pathologists, pulmonologists, radiation oncologists, as well as our thoracic radiologists. And they each have their own areas of specialty within thoracic oncology. And because of our extensive focus on a particular range of cancers, we are really the experts in the area when it comes to lung cancer and provide patients with individualized treatment recommendations. And that gives them not only the best prognosis, but we really strive for quality of life improvements. And these really have to be coupled very well together. At Moffitt, we focus not only on learning on our clinical observations, our clinical research, as well as our bench research to improve treatment outcomes, as well as the treatment advances, and bringing these to the patient's bedside. What do we think about impact, though? Ultimately, that's what we're going for here. And this includes, as well as improving outcomes for a particular patient, but more globally to change standard of care and FDA approvals. And you're going to hear more about from Dr. Saltos later on today about how we do this. But what does it mean for our patients? It means that we have to have an understanding of the unique characteristics of their individual cancer and really to develop a personalized treatment plan in this setting. And thus, we really have this robust clinical trials program at Moffitt that focuses on thoracic malignancies. Again, we're looking at advancing the advancements in the field and the treatments that not only can be found at our cancer center primary site, but also working to get these out at our satellite sites. And so later tonight, again, you'll hear a little bit more about this as we work to share our exciting breakthroughs throughout the evening. And this will start with our first speaker. I do want to bring your attention to the functions at the bottom of your screen there that are hopefully there. There is a Q&A button that you should be able to see at the bottom. We would love to hear from you. I know our speakers and experts would love to hear from you. And I think this will provide you an opportunity to get those questions that you want answered by us. And I invite you to just go ahead and type your questions in throughout the chat and throughout the talk here. So now we'll move to our first presentation. So one of the emerging areas of study is really within the realm of precision medicine, and it is utilizing liquid biopsies to help understand any given patient's cancer on an individual level, okay? And this will help us not only with diagnosis, but also throughout the patient continuum. And with that, I'm pleased to introduce Dr. Bruna Pellini. She is our assistant member of Moffitt Cancer Center in the Department of Thoracic Oncology, a clinical investigator who will speak to you more about our liquid biopsy program as well as our research. Dr. Pellini?
Thank you, Dr. Gray, and good evening. I'm so honored to be here and talk to you guys about a topic that I'm so passionate about, and that is my research focus. So, first of all, lung cancer. When we talk about it, and when I discuss this with my patients, I always explain that there are two big subtypes. So we have small cell lung cancer. That's a little bit more aggressive type of cancer, is less prevalent. Around 13% of patients present with that. And then we have the big portion of lung cancer that consists of non-small cell lung cancer. When we talk about non-small cell, when the pathologists look at the cells under the microscope, they are able to differentiate what specific type of non-small cell lung cancer it is. So we have adenocarcinoma, which is the most common subtype. Then we have squamous cell carcinoma, that is the second most common subtype, and we have large cell, that is a little more rare. And then I put an in-between category that is the neuroendocrine tumors because they are not non-small cell and they're not small cell. So their categories right there in between. So I will focus today a little bit more in non-small cell lung cancer since it's the most common subtype. So again, going back to the, I'm sorry, let me go back one slide. Going back to the distribution of lung cancer, we have adenocarcinoma. You can see in this pie chart, approximately 55% of the patients have that. Then around a third has squamous cell carcinoma, and 11% of patients have other subtypes. When you look at this pie chart, you may think, well, although there's some distribution, it all looks like the same disease, but it's not. Because what we see is that lung cancer nowadays, we have so many subtypes accordingly to the genetic alterations. I'm sorry, I'm having technical difficulties with the slides. So there's so many subtypes accordingly to the genetic alterations. So for a person to develop cancer, the genes need to get messed up. And then a gene that gets altered can develop cancer growth. Nowadays, we have amazing technologies to identify those genes that are altered. And as you can see here, we have around a third of patients with non-small cell lung cancer that has EGFR mutations. We have tablets, we have targeted therapies, pills that we can give to patients with that kind of alteration. Not all of the alterations in EGFR genes, but what we call EGFR sensitizing mutations. Then around a third of patients has KRAS mutations. There's a couple of drugs that have been have shown to have good activity for a subtype of KRAS mutations, but we're a long way to be able to treat very well and in a personalized way patients with other KRAS mutations. And then you have all the small parts of the pie that have other alterations like BRAF mutations, ROS1 fusions, ALK rearrangements. And for some of those alterations, we do have targeted therapies. We do have pills that we can treat patients with. But we are far away from having one pill or one targeted therapy for each gene. And that's the role of clinical research. Now, when it comes to liquid biopsies, why is that important and what is that? So what is a liquid biopsy? A liquid biopsy is basically a clinical test or assay that is performed on a sample of body fluid for cancer assessment. This can be done in blood, plasma, or it can be done in urine and other biospecimens depending on the cancer. So the majority of the assays that we have nowadays, the majority of the tests that we have, they use blood as a source of material. So how are we able to detect these mutations in blood? So when patients have cancer, the cancer cells, they shed DNA into the bloodstream. In the bloodstream, you're going to have DNA coming from normal cells and coming from cancer cells. How are we able to differentiate what is normal and what is not? Well, the mutations that happen in lung cancer, we are aware of them. And we're able to, through a lab test, to kind of pull the DNA that has the cancer mutations, and we're able to identify. Now, this is not a perfect test, obviously, because it depends how much DNA the cancer cells are shedding at the time for us to be able to detect it. If we do detect, that's great. We can treat patients accordingly to those results. If we don't detect, then at those times, we still may need to pursue a genetic testing on a tissue biopsy. So why are they important? I kind of already went over this. So first of all, we want to know if a person has any of those mutations that we can offer targeted therapy. Because sometimes I have patients asking me, Dr. Pellini, can I get immunotherapy? I was like, well, you may, but if you have these mutations that we have great pills that work very well, we want to know, and that's the gold standard. That's the way we should treat you. Also, not all patients have this cancer tissue, this good biopsy that we can do this genetic testing on the tissue. For instance, this past 2 weeks, I saw 4 patients with stage 4 lung cancer that presented with fluid within their chest, pleural effusions that we call, and that doesn't have a lot of cancer cells. So those are the patients that I need to rely on the liquid biopsies to kind of look for those mutations that are so important for us to decide on treatment. And also, because of that tool that we have, these liquid biopsies, we can personalize treatment because a lung cancer treatment is not one size fits all. Now we can personalize and engineer the best way to treat our patients. But one important thing, liquid biopsies, they do not replace tissue biopsies because tissue biopsies are able to tell us this is lung cancer and what type of lung cancer. So it's diagnostic. Liquid biopsies, they give us mutation information, so genetic alterations, mutations, in order for us to personalize treatment. So when can we use those liquid biopsies? We may use those nowadays as of April 2021 for patients with advanced non-small cell lung cancer or patients with metastatic lung cancer. And there are 2 FDA-approved liquid biopsies. One is Guardant 360 and one is Foundation One Liquid. So what is the difference? Guardant 360 has less genes in the panel, so it's looking for mutations in the important genes that we have already approved therapies, but the panel of genes is not that broad. But the results come back a little faster in 7 to 9 days. And because of the issue that I told you that it depends how much DNA from the cancer cells is shedding to the bloodstream, this assay has a very good limit of detection, meaning it doesn't require a lot of DNA from the cancer cells to be able to detect a mutation. Now, Foundation One Liquid CDX is also a very good assay. It has more genes. It has over 300 genes that it looks for mutations. The results take a little bit longer, and the limit of detection is likely lower. So if you have a mutation that is present in very, very low levels, it may not be able to catch it. So what are we doing here at Moffitt regarding these new technologies? So there's a few research projects that we're working on. One that is going to open soon is it's called Thoracic Trial Selection Protocol, and I invite you to be part of it. And this way, we're trying to streamline the enrollment of patients in clinical trials that still require biopsies from tissue. We're also going to be collecting blood to kind of track how patients react to different therapies. We probably will be opening a prospective blood collection study as well for patients that are not eligible for clinical trials but want to participate and have their blood assessed for these different mutations. And here with my lab mentor and with mentorship by Dr. Gray, I'm developing a few new technologies for us to detect a cancer when it's in early stage. And the hope is that with these technologies that we are developing, we can detect the disease before it shows up in the CAT scan. And this way we can escalate or de-escalate treatment for patients undergoing surgical treatment. Because sometimes there's patients that appear in my office and they say, Dr. Pellini, I had stage 1 lung cancer a year ago. How come I have stage 4? It's because CAT scans are great, but they're not perfect. So we need different tools to kind of enhance this cancer detection before it appears on the CAT scan. And also another way that I see this technology going is if it's successful, we could even potentially use in the future for lung cancer screening with CAT scans. And for our patients with stage 4 disease, we also have clinical trials that are incorporating these liquid biopsies to personalize treatment. And we'll be opening one for stage 4 disease and another one for resectable lung cancer. I invite you all to participate. And if you're interested in any of these technologies in supporting our research, you can always contact Carrie and our team. We'll be very happy to have your support. Thank you very much.
Outstanding presentation, Dr. Pellini. Thank you very much. She is truly a superstar, and we were successful in recruiting her from WashU. Talk about an area of expertise, excellence in precision oncology, and just bringing that expertise here to really advance things for our patients, not only within the Tampa Bay area, but across the state of Florida. as we look at getting more information regarding these personalized treatments, it's really going to be critical, like Dr. Pellini mentioned, that we get these results, we get them in a timely fashion. And if there are opportunities to do this in a non or minimally invasive way, it is very helpful to help move the field forward and can help us develop not only the best treatment standards for our patients, but also beyond standard of care to look at that clinical trial, that novel therapy that has maybe not made it yet to the clinic for standard of care, but we have available at our site. And so it is a critical piece of building out our research infrastructure. Thank you again for that excellent summary. And so now we'll transition to Dr. Saltos. If Dr. Pellini is our superstar, Dr. Saltos is our rock star. He is another clinical investigator in the Department of Thoracic Oncology and an assistant member here. He leads up our clinical trial portfolio. He's our clinical research medical director, not a small feat. We're one of the largest programs at Moffitt that focuses on clinical research and helps to run a trial portfolio of 30 to 40 and supervise a team of over 25 to make all of this possible and bring that research to the patient, to the bedside. And so with that, I will hand it over to Dr. Saltos to talk to us about precision medicine. Dr. Saltos?
Thank you, Dr. Gray. Thank you. Happy to be here tonight and to highlight our research program and how this ties into precision medicine and the idea that we'll find the right treatment for the right patient and how clinical research that we do here ties into that. So I want to start by just highlighting the fact that there was a fair amount of media attention in the past year about some improvements that we've seen in cancer survival. Studies that were conducted both by the National Cancer Institute and the American Cancer Society showed a historic drop in the cancer death rate in 2020. And both of these research studies looked at more granular data, and they found that this was in large part attributed to an improvement in lung cancer survival. And furthermore, the improvements in survival was in large part because of better treatment. And I think this is a reflection of what we've seen, as medical oncologists, being able to offer much better treatments than we could 10 years ago. Specifically, these precision treatments based on the molecular makeup of each patient's cancer, as well as immunotherapy treatments, which have really also represented a major breakthrough. And so at Moffitt, we make it a mission to participate in clinical research to help to find and advance some more better treatments for our patients, because it still remains a fact that lung cancer is unfortunately the #1 cause of cancer mortality in the US and in the world. And we have a long way to go to find better treatments. And we see this every day where we are looking for opportunities to offer new treatments for our patients. And so Moffitt is one of a select group of cancer centers in the US that are designated as NCI, or National Cancer Institute designated comprehensive cancer centers. What that means is that we have a unique infrastructure of basic science research resources, outreach, and a number of other interdisciplinary staff and teams that can help us to collaboratively, comprehensively tackle cancer from more than one angle. And within Moffitt, the thoracic oncology is a strength. We have what's known as the Lung Cancer Center of Excellence. And this is a specific body of researchers and collaborators who specialize in lung cancer. And there are members of this Center of Excellence who span from clinical clinicians, such as ourselves speaking tonight, but also researchers who do discovery of fundamental aspects of how lung cancer works and what treatments might work better, as well as those who work with cancer outreach, as well as screening, detection, and even smoking cessation. And so we all work collaboratively, and that provides us a strength and a way that we can advance cancer research in a unique way. Specifically speaking to our clinical research program in our thoracic department, we have a large team, more than 25 staff members who are here specifically to work with clinical trials. And this is on top of the nurses, the providers, the clinicians in the clinic who you might encounter as a patient as part of your clinical care or participation in the research trial. At any given time, we offer anywhere from 30 to 40 unique research trials for patients, and this sort of is a representative of the fact that lung cancer is not just one disease. There are many different molecular subtypes of lung cancer, aside from the differences between small cell and non-small cell and other thoracic malignancies like mesothelioma and thymoma. About half of the patients that we accrue to clinical trials here in our thoracic department are participating in what are called investigator-initiated trials, or IITs for short. These are studies that were designed and led by Moffitt, and these are really mostly coming from research ideas that start from our team. These are a complex undertaking from the concept to execution, and it's made possible through basic and translational discoveries that we have from our partners in the science departments at Moffitt, the huge infrastructure that we have and the efforts that go into making sure we have the staff, the space, and the technical means to carry out these research studies, not only reliably but also safely. And it also requires funding support. There's a lot of efforts that go into these research studies, and funding, cancer funding and research funding is always a high need. So I want to take the time to highlight one specific example of an investigator-initiated study that we have ongoing here at Moffitt. This is a research study looking at two cancer drugs, pembrolizumab, which is an immunotherapy, and vorinostat, which is what's known as a HDAC, or histone deacetylase inhibitor. It's an oral anti-cancer molecule. So this research trial started as an idea from basic research that was conducted at Moffitt, and there was a discovery that this combination of drugs seemed to uniquely enable the immune system to better attack cancer cells in the lab, and that translated from a lab research idea into a clinical trial through a lot of the efforts in our thoracic department. And to date, we have had a measure of success with this trial in that we've accrued over 100 patients, and we are very thankful to those patients, volunteers, and their families who have participated in this study. And we have been able to demonstrate some promising outcomes, and also, more importantly, for those patients who've had a response on this treatment, it offers a hope for those patients. And I'm happy to report that at least a preliminary analysis of our patients treated to date on this research study has shown approximately twice as many patients who were given this combination had a major tumor shrinkage compared to patients who were given pembrolizumab immunotherapy by itself, which is represented as a standard of care currently in lung cancer. And based on what we've seen on this research study and some of the efforts to collect more data on the patients who are on this study, further research is planned to learn more about how and why this treatment may work in some patients and maybe not in others. And so we can select where this would be the best to tailor this treatment for which patients. And also to hopefully be able to bring this approach of treatment to more patients in the future. I also want to highlight that, again, that human impact that the individual patient that might experience through the research that we do, and that the research efforts aren't just science necessarily, but it also affects people. And so I know there are many examples like this. I can just think of one example of a patient of mine who I met a few years ago. We'll just call her Jane for purposes of this example. But so she had a diagnosis of advanced lung cancer, but it was surgically removable back when I first met 3 years ago, and underwent surgery and chemotherapy, and there was a sigh of relief that hopefully things had gone well at that point. But unfortunately, about a year later, we discovered that the cancer had returned and had metastasized, and it was very devastating news, obviously. So looking into her treatment and her tumor profile, we found that she didn't have any of the mutations, as Dr. Pellini had highlighted, that might open up the door for a targeted TIL therapy. Also, it turned out her standard profile showed it may be unlikely for her to benefit from immunotherapy. But there was a personalized medicine panel of many genes that showed she had what's called a high tumor mutation burden. And this suggested that a novel combination of immunotherapy might be beneficial. And so she was enrolled on another clinical trial. This is another investigator-initiated trial looking at a combination of nivolumab, ipilimumab, and nintedanib. And the patient had a wonderful response. So Jane is doing very well now, almost 2 years later, with what is essentially a complete response, meaning her tumors that had metastasized that we discovered are no longer visible on her scan. And she's not only had a great response to treatment, but also doing well and returning to all the activities of her normal life at this point. And so, of course, we hope that this continues, and it just is a testament to how finding the right treatment for the right patient based on their specific tumor profile can offer benefits, and how the fact that we have research studies looking at these specific treatments can really impact patients.
Absolutely wonderful. Thank you for that excellent presentation, Dr. Saltos. Really at the cutting edge of where we want to be and where we are going. So I wanted to just take a moment, and I'm reflecting a little bit as I listen to that story, and I really appreciate you sharing that with us, Dr. Saltos. I think it brings us back to what our foundation is at the thoracic department. It really has to do with generating those best patient outcomes, hearing more of those stories that you describe, and being at the front edge of where we are with therapy and with research innovations and driving that forward. It made me think about a patient that I met over 8 years ago now. Unfortunately, he came and he was referred on hospice. He had a young child at home, and he had a 10 centimeter tumor in his chest. I think I was the third physician that he had seen at this point. And everyone that he'd seen prior to me was really recommending again that he go on hospice. But besides his tumor, and that it just happened to grow in not the best place. It was growing near his blood vessels, had actually cut off the blood supply and clotted off, caused some clots and backup of his vasculature, and caused his arm to swell quite significantly, honestly, when I met him. And we just started talking and just listening to him about, what is your day like? What are you able to do? And I was like, we have this new exciting agent. It's called an immune checkpoint inhibitor that targeted PD-1, PD-L1 at the time. And it was a kind of first in human trial of this agent that a lot of people now know as nivolumab or Opdivo. And he ended up being able to get him, and we enrolled him on this first trial. He got one of the last slots on there. This trial that's now published in New England Journal led to FDA approvals and really has changed the paradigm as well as the standard of care for us. And he, needless to say, he's still alive today. His daughter is now in high school. And it's just very joyous to see that we can make a difference. And it is really on us, including especially those on this call, to act not only as guardians, but also stewards, to move these initiatives forward for our patients. It's really critical toward reaching our mission, where every patient is an individual. How do we get there? And how do we get more stories like this, right? First, we have to take that pause. We saw that nice pie chart, those pie charts. There are different types of thoracic malignancies. Not all of them are lung cancer. And even within lung cancer, while it is the majority, there's subtypes of different types of lung cancer. And then we really want to drill down to that molecular and DNA level to say, what is really going on? The science, clinical science, the bench science needs to drive us to have better understandings across the spectrum of the subtypes of thoracic malignancies. And to do this, we need to invest in seed funding and to be good stewards, as I mentioned before, and then also look at where we direct that funding, and that should really be toward where we have the biggest impact. And at this time, I wanted to take a moment, and I'd like to just introduce you to one of my partners here, our partners at Moffitt, Ms. Carrie Bean, and she's with the Moffitt Foundation, and she partners with us here in the Department of Thoracic Oncology to ensure that when we receive research dollars, that we really get this directed to the right place, to clinical research, to get things off the ground and make advances for patients toward cancer treatments and to reach our patients. And if you'd like to learn more, I highly encourage you to please contact Carrie. I'd really love it, Carrie, if you'd be able to say a few words, if that's okay with you.
Yeah. Thank you so much. Thanks, Dr. Gray, for including me this evening. And thank you all so much for joining us. I hope what you've learned so far has been informative. And I'm also really excited for us to have quite a bit of time for question and answer and just an open panel with these amazing speakers. One of the things that I took away from what you all shared is just how transformational some of the research that is happening here in thoracic oncology is. And I have the pleasure of getting to work with Dr. Saltos, Pellini, Gray, and the rest of the oncologists and surgeons in the thoracic department to understand what they're working on, what their teams are working on, what research they're excited about, and what they see as coming next as a team. And once I do that, I'm also available to each of you all if you're interested in learning more or you're interested in a specific area. We can partner together to help make an impact and a change in that specific mutation or component of thoracic treatment and care. So I am the only thing standing between the Q&A, so I am going to step away so we can continue to visit and hear from these speakers while we have them for this extended amount of time. But my confirmation is up there and you all can, my information, excuse me, and you all can reach out to me if you need anything.
Thank you so very much, Carrie, and we appreciate all that philanthropy does to make our research possible. And now, ladies and gentlemen, I invite you to the Q&A session. We'll get that kicked off. We have a lot of questions already posted, so we'll get this started here. So one of the questions came up here is really directed, a couple of them, I'll try to ping these together, I think, and look at themes, okay? So one of them pertains to ALK rearranged lung cancer, wanting to hear what we have as far as research, ongoing planned research, as well as what do patients need to look out for from a side effect profile when we think about utilizing these ALK inhibitors. Dr. Pellini, do you want to tackle that one first?
Sure. So regarding studies, from the top of my head, I can remember that for patients that undergo surgery, if they have an ALK rearrangement, we're offering targeted therapy in the adjuvant setting, meaning after surgery, because now the standard of care is chemotherapy. But since these drugs work so well in stage 4 lung cancer, why not try them in patients that we can achieve a cure, right? So that's one of them. In the metastatic setting, I do not recall anything frontline. Maybe Andreas can help me regarding something for second line and beyond. In terms of side effects, these drugs are great, but of course they have some side effects, unfortunately. If we could come up with drugs that had no side effects, I think we would get a Nobel Prize. But having said that, some of the side effects from these agents are increasing liver enzymes. So we always looked at the blood work. This is not something a patient will feel, but if the liver enzymes are going up, then we have to decrease dose. Sometimes we have to hold for a while. Fatigue is something that happens with these drugs as well. again, we can do those changes. Kidney function may impair during the treatment. We always watch out for this, but patients wouldn't feel anything, particularly regarding that. Swelling sometimes happens with some of those drugs. Many times, unfortunately, diuretics, water pills, not necessarily work, so it depends where the swelling is. There are other things that can happen, like nausea, vomiting, or constipation, and we have medications to treat those. So I would encourage you to talk to your physician and say exactly what you're experiencing, and there's some changes that we can make so you can feel better. Fantastic.
Dr. Saltos, I didn't know if you wanted to add to that, because I know, again, you head up our clinical trial portfolio, and just so everybody understands, it's not just about a one-time portfolio. It's really about also looking ahead and seeing where are we going, what's coming down the pipe, and how do we keep on top of that and make sure that these agents are available to our patients, all of them that walk in the door, and we can match you to the best therapy. Dr. Saltos?
Right, thank you. So on the topic of ALK specifically, ALK-fused lung cancer, we would at this moment recommend standard oral therapy, tyrosine kinase inhibitor therapy, such as alectinib, or there are others that are also maybe appropriate in the first line. We do have a number of clinical trials that a patient who may be progressing after such treatment may be eligible for. These currently are trials that are open to many lung cancer patients. We specifically have two trials that I can think of that will be in the works, that will be specifically looking at patients with ALK fusion who have progressed on initial therapy. One with a combination ALK with a novel drug to increase sensitization, and another looking at an antibody drug conjugate. But these will be coming in the future. I think it's also important to note that if patients have progression after taking an oral therapy like ALK or other oral targeted therapies, it's very important to obtain either a new biopsy or liquid biopsy to determine why the cancer became resistant to the therapy. Because in many cases, not all cases, in some cases, a new tailored therapy can be directed based on a new genetic mutation.
I think that's a fantastic answer. And I think it's actually, there's a great segue here for this. So there's a patient here, there's an individual here whose mom is on the Tagrisso ramucirumab study, for which Dr. Saltos is the principal investigator for, and just recently presented early data from that study at our World Congress on Lung Cancer. So you have the world expert here on this. It's fantastic. So good to have him with this question. They had a EGFR mutation, her mom did, and she had a biopsy about a year ago. And the question here is, how do you make that differentiation between when you need a biopsy for tissue and when do you need a liquid biopsy, and how do you utilize that? I know Dr. Pellini touched on this also, but I think Dr. Saltos, we'll start with you because I know you're leading this trial, and then maybe we'll go to Dr. Pellini.
Sure, that's a great question. And I think in some ways, first of all, it's important that when a patient is progressing on therapy is when to obtain these biopsies, whether tissue or liquid biopsy. If the patient is currently doing well on the anti-cancer therapy with no signs of cancer growth, a biopsy may be of low utility, or especially a liquid biopsy could be of lower utility when there's no sign of active cancer. But at the time of cancer progression, I think the gold standard, what we had always considered being the best, had been a tissue biopsy. And I think that still, for the most part, holds true. There is potentially the idea that in some cases, one tumor might have different characteristics. Say, if there was a metastasis from the cancer in the liver and a different metastasis from the cancer in the lung, potentially they could have different molecular characteristics. And the advantage, one advantage that a liquid biopsy might offer is that you might be able to simultaneously detect any alterations that are present at either tumor site without missing the other one. But at the same time, a tissue biopsy probably has some somewhat better reliability for the specific test of what you biopsy. So I think the short answer is they both have an important role, and I think they should both be discussed with your oncologist at the time if such an event occurs, if your cancer is progressing.
Perfect. And Dr. Pellini, did you want to expand on that? And then I have a subsequent question that follows along those lines that has to do with what are those risks that come with doing a biopsy? What about, will it spread my tumor?
Can you repeat the last question, Dr. Gray? I didn't hear you very well.
So the question kind of builds on top of what Dr. Saltos was just talking about, and taking if I have to undergo a tissue biopsy, does this put me at risk for having my tumor spread? What are those side effects, on top of, how do you make that decision about when to do this, when to get the liquid versus the tissue?
Mm-hmm. So for EGFR mutant lung cancer, since this is the topic, I would encourage doing a tissue biopsy for one reason, that sometimes the cancer type changes after therapy, meaning that a patient that had adenocarcinoma, that is a type of non-small cell lung cancer, may change to a small cell lung cancer, and in some rare instances, a squamous cell carcinoma. And the only way for us to know that is to do a tissue biopsy, right? In terms of the genetic material changes, I just like Dr. Saltos explained beautifully, the liquid biopsy sometimes catches these gene alterations from different cancer lesions that we see on the CAT scan, whereas if you only rely on that tissue biopsy, you may just see that mutation in that specific lesion, right? So I think they complement each other to some extent. Of course, we don't want to put our patients through repeated biopsies over the course of years, and that's why there's an advantage to doing liquid biopsies if we're planning on doing that multiple times. Now, in terms of the cancer spreading with a biopsy, that's not true for lung cancer, so a patient shouldn't be concerned about it. A biopsy nowadays is a very it's invasive, but it's not that invasive like a surgical procedure, and they do CT-guided biopsies. So is the interventional radiologist doing that, or an interventional pulmonologist? So they're very precise on the way they retrieve tissue. That will not make your stage 1 go to a stage 4. It will not make your stage 4 progress. So do not worry about that. That's the way to go. So I think that's important to know. And we need that for diagnosis. Again, liquid biopsies do not diagnose cancer. They just tell us about the genetic alterations in the cancer.
Fantastic. I think those are great, excellent summaries. So Dr. Saltos, I'm going to ping you for the next question. It really has to do with clinical trials. What makes somebody eligible for a clinical trial? Can you speak a little bit about that process and things that are looked at?
Sure, that's something that comes up, certainly anytime a clinical trial is discussed with a patient, the whole process is certainly, it's a mystery unless you've been through it. So every clinical trial that we offer has what's called a set of eligibility criteria. And essentially, there are specific population of patients who are being recruited for the research study. And the studies that we have to offer for our patients may either be recruiting a specific type of lung cancer, or they may be more broad and say non-small cell lung cancer altogether, or they could be more narrowly focused to those who have a specific mutation. In some cases, a clinical trial may be open to a patient regardless of what prior treatment they've had, or in some cases that may require the patient not have had prior treatment, or that they have had prior treatment. And so your oncologist would gear which trial might be applicable to that requirements, and they would narrow down which clinical trials would be applicable to you as a patient. In addition to those major criteria, there are many safety-related criteria for research studies related to organ function, such as cardiac health, liver function, and kidney function. And it's important for safety reasons because these drugs might affect those organs or be metabolized through those organs in the body, that participants in the research study have otherwise healthy organ function, for example. And there might be other specific criteria where a medical condition might mean a particular treatment may have an elevated risk, and those patients may not be eligible to participate in the research study, again, for reasons of safety. And the first, one of the biggest concerns that drive these criteria is the safety and welfare of patients. And so it's important to know that paramount to everything that we do in clinical research is the safety of our participants and making sure that we are offering treatments that are appropriate and safe in knowing that the interventions may have an uncharacterized side effect profile in some cases, but once patients go through a screening procedure where they would have organ functions tested, such as getting blood work, getting an EKG, and having CT scans, and sometimes a biopsy of a tumor through the screening process, you would be able to find out if you meet all the criteria. And if they do, then patients would be scheduled to start their treatment on the study.
Yeah, it's a very, I think what really resonated when you were talking is that it looks like a lot of hoops, so to speak, to have to jump through to get on these clinical trials, but safety is at the forefront, and also ensuring that we match you to the right therapy at the right time along your patient care continuum. And that is very important and critical to not only giving you the best result possible, but also advancing the field. Dr. Pellini, there's a question here about looking at small cell lung cancer. What do you think's next? What's upcoming? What's on the horizon? And then Dr. Saltos, there's a similar question on meso, which I'll direct to you subsequently. Start with Dr. Pellini.
So first, thank you. So for small cell, we have, I think currently one study open in the frontline setting that we're combining different therapies, but I have to be honest, chemotherapy is the backbone, and I don't think that's gonna change for first line setting because small cell lung cancer responds very, very well to chemotherapy. So we don't want to take away that great response. In terms of second line and beyond, we have a couple of trials open. One is actually after patients get the chemotherapy and the immunotherapy, which both are standard of care. The patients can get some chest radiation and then get a pill plus the immunotherapy to see if that enhances the activity of the immunotherapy after patients undergo this first phase that you get chemo and immunotherapy. For second line, we have a study combining immunotherapy with a new drug that was just recently approved that is called lurbinectedin. And that's a very good study, in my opinion. And for third line, I do not think of anything from the top of my head, but those are the two that I know for sure are accruing and they're good options.
Fantastic. And Dr. Saltos, you want to tackle that question on mesothelioma? You know, they're talking about treatments. They're also asking about biomarkers. What should they be on the lookout for?
Sure. So I think one of the most recent kind of exciting advances in mesothelioma was the FDA approval of immunotherapy for patients with mesothelioma. And so this is kind of a recent breakthrough. We've been participating in research studies giving immunotherapy to patients with mesothelioma for some time now. We have plans on beginning a research trial, hopefully soon, that's looking at the possibility of combining chemotherapy with immunotherapy, sort of building upon what we've seen in non-small cell lung cancer, where, you know, prior to immunotherapy, chemotherapy had been the only available standard treatments for most patients, and then immunotherapy was usually given separately from chemo, but now it's very common for us to give chemotherapy and immunotherapy together in non-small cell lung cancer. And so I think that's a logical step for us to be testing in mesothelioma. We also have other immunotherapy research studies going on for mesothelioma, and I think we're always on the lookout for additional clinical trials, and mesothelioma is an area of high interest. We do have a number of research studies say that are in our pipeline of research that say are looking at molecular alterations or genetic factors in various tumors. One that comes to mind is one that's looking at mesothelioma in addition to other tumors that have alterations in CDKN2A, which is a proliferation gene in cancer. And so the needle is moving also in mesothelioma. I would say, it being a rare disease, it certainly represents a challenge to have as many research studies in a rare cancer like mesothelioma, but we do our best to make sure that we try to offer as many research studies as possible and keep our care on the cutting edge.
Yeah, I think those are really fantastic points. I think what you've heard here so far described by Dr. Pellini and Dr. Saltos is that sometimes this research, it comes from interrogating, cancer cells in a lab, for example, but it also comes from these clinical observations and having an understanding of what are the needs within any given patient population, and that their expertise is helping to guide that research and navigate it forward so we can expand upon the treatment armamentarium for every patient at the right time. There's really some interesting questions here that are coming up also along the lines of EGFR mutant lung cancer, and would love to hear from Dr. Pellini. If I get a biopsy and get my molecular testing, liquid or tissue, and I have an EGFR mutation, what can I do? And then on top of that, not just in the stage 4 setting, but there's also, what should I expect with the use of the EGFR inhibitors in the adjuvant setting? You can talk on those fronts.
Okay, so we actually have 2 trials for first-line treatment for EGFR mutant lung cancer. One is combining with chemotherapy. That we have to look back at the past. Like we know chemo works very well for EGFR mutant lung cancer. We know the pills work very well. They work better than chemo when compared head to head. So what if we combine them two, and how long we'll be able to contain the disease? It will be longer than the pill alone, will be tolerable. So this is one accruing trial. Second one is the osimertinib, that is Tagrisso, with Cyramza, that is ramucirumab, is also a very good option for patients. If you're not too fond of getting chemotherapy, potentially getting chemotherapy, that's an option. So those are 2 of the studies we have for first line. Second line, we currently have one study open that is a multi-center study that is testing immunotherapy combined with chemotherapy, because like I told you, EGFR mutant lung cancer does respond to chemotherapy. It is an important line of therapy. Now the question is, does immunotherapy work better when we add on, or it doesn't? So this study is investigating that. And we are about to open another study for second line and beyond for EGFR mutant lung cancer. So we'll have plenty of options. For patients undergoing surgery that have the tumor has an EGFR mutation, Tagrisso is approved, and the outcomes regarding disease coming back, they are the ones that we have. We don't have data on survival, meaning we don't know if giving this pill will make patients live longer. However, we know that the odds of the disease not coming back at certain time points is much higher if we give the pill. For instance, if you have a stage 2 disease, if you get the pill, there's 91% chance that you won't have a disease recurrence at 2 years versus 56% chance of having the disease come back at 2 years. So it's very different, right? So although we don't know in terms of survival, we are assuming that we'll have a benefit in survival, but only time will tell us.
Fantastic. And Dr. Saltos, I think we're getting close to time here. But one of these questions here really resonated with me. They asked, how do you determine which type of cancer to research? How do you choose?
That's a great question, and I think we would love to make sure we had research and we knew everything about every kind of cancer, and we try as best we can to make sure we have research, trial opportunities for any patient that walks in the door at Moffitt. Now, given the broad diversity of cancer and how, if you really divide cancer into all of its subtypes, there can be hundreds and hundreds of different scenarios or types of cancer. We at least make sure we make it a priority to have research studies for those where we see the most patients. But that's not our only consideration. There are some cancers which are, for example, called orphan diseases or very rare diseases. One example that comes to mind is thymic carcinoma. The thymus is a gland that sits in the upper chest, which helps produce immune cells in our childhood. And it usually degenerates or atrophies and it doesn't serve much of a function as an adult. But the thymus tissue can develop a cancer really at any age. And thymic carcinoma represents the most aggressive type of thymic cancer. It's very rare, and there are very few treatment options. We have recently opened at least one study for patients who were diagnosed with thymic cancer, and we're negotiating opening another research study for these patients. So even though we don't see very many of these patients, given the fact that they don't have very good alternative options, that's become an area we want to target. We want to make it a possibility that we can offer hope to patients with common cancers as well as those who have maybe rare cancers which have very little bit known about them.
And I think once kind of a theme I'm seeing here also in both the chat as well as in the Q&A is what did it describe as, okay, so I'm on a clinical trial now, I'm doing really well. What do I need to do to sustain that kind of response? And if that drug stops working for me, what's next? Dr. Saltos, do you want to talk about that?
Sure. So I think for one thing, I think it's hard there's always the motivation to want to make sure you can control everything you can. You're doing everything right. And that's a very healthy impulse. It's sometimes it's also want to make sure it's clear that when the cancer does progress, it's not a reflection of necessarily anything the patient does. But that being said, there are some small things I think are maybe not so small, but that we've seen that can help patients. One of which is attention to physical activity. I think we have some members in our thoracic program, especially Dr. Robinson, if you've worked with him. He's a very big proponent, and I'm a big believer too, in exercise for patients with lung cancer. And I think there are a number of research studies across all types of cancer that not necessarily intensive exercise, but low intensity aerobic exercise, such as walking every day, can really have an impact in survival and treatment outcomes in cancer patients. So I think what I tell patients in my clinic is not necessarily that you need to feel you have to take a certain amount of steps a day or do a certain specific gym routine, but I wouldn't limit yourself in terms of exercise. If you feel up to going for a walk, it's encouraged. You don't necessarily need to rest because you have cancer or you're undergoing cancer treatment. So exercise is encouraged. And I think attention to your general health, and that goes from everything from nutrition to mental health, making sure you take time to rest, take time to reflect, and whether that's spiritual care, whether that's mental health care, whether that's family, try as much as you can to focus on your life, and because the hope is, at least for some of our patients, and these treatments are very effective and sustained. And I think it's healthy to have the attitude of, why shouldn't that be you?
Fantastic. So thank you very much, Dr. Saltos and Dr. Pellini. I think that will do it for us tonight. I do want to remind everyone on here, thank you for joining us again. I really hope that you found this webinar informative and looking at what we are doing to revolutionize cancer care and provide the best possible outcomes. And we really appreciate your continued support. And for those who posted additional questions that we were unable to get to tonight, I assure you that we will get back to you on those. And have a great evening, everyone. Thanks again.